This video is all about the 2 steps of photosynthesis. The goal of photosynthesis is to use sunlight to make sugars. This occurs in 2 steps, the light reactions and the dark reactions. The light reactions require light. They also will use water. And during the light reactions, oxygen gas is produced as waste, and then 2 energy-storing molecules are made, ATP and NADPH. Those energy-storing molecules will then get used in the dark reactions along with carbon dioxide gas. The dark reactions is essentially using the energy from these molecules to glue together carbon atoms in hopes of producing glucose. That glucose would then get broken down by the plant, either immediately or later, for energy. So plants will still do cellular respiration to break down that glucose. This is going to be kind of intense for the light reactions because you are expected to know a lot of details for the light reactions. The light reactions involve the thylakoid. So we are inside a plant cell, inside the chloroplast, inside these little stacks called thylakoids. The molecules that absorb light in the thylakoid are called pigments. So light will come in and excite an electron. In plants, there are 2 photosystems, photosystem 2 and photosystem 1. Unfortunately, they were discovered in the reverse order and nobody has renamed them. So photosystem 2 actually comes first because it was discovered second. But it is used first in the plant, and then photosystem 1 is used. These 2 photosystems best absorb 680 or 700 nanometers of light based on the pigments, based on the chlorophyll that's inside of them. Those pigments of light are red. So most plants best use red light. All right, here we go, the light reactions. This is an intricate process for which I will draw a diagram and then I will show you some pre-made photos on the coming slides. So the light reactions occur first when light hits a photosystem. The first photosystem that's used is called photosystem 2. There is another photosystem that's used later that's called photosystem 1. Photosystem 2 and 1 are made of a series of pigments. Those pigments will absorb light energy. The first photosystem best absorbs light energy at a nanometer of 680. And the other one best at 700. So that's just the wavelength of light that works best for those particular pigments. So what happens is a photon of light from the sun comes into that photosystem. In those pigments, an electron then gets excited because the electron gains energy from the light. That electron gets excited and it gets grabbed by a protein that's called the primary acceptor, meaning it's the first protein to accept that excited electron. That primary acceptor then passes the electron down an electron transport chain. So this is a similar series of proteins as which we saw with cellular respiration. Same idea. We've got proteins on each of these steps. Those proteins will pass the electron down. As the electron is falling down that chain, it loses a small amount of energy. So this is the same idea that we saw in respiration. where there is energy released from the falling electron. Since that photosystem had an electron that then got excited, grabbed by a protein, and passed down the chain, eventually that photosystem would run out of electrons because the primary acceptor keeps taking them. However, there is a replenishing supply of electrons such that those pigments in the photosystem will not run out of energy. And the molecule that provides the electron is water. So plants use water for a variety of things, but one of them is to supply the electrons. So that water will resupply the electrons so that the photosystem does not run out of electrons. During that process, hydrogen ions are produced and also oxygen gas. Those came from the water molecule. That oxygen gas is trash, so the plant releases that. So that oxygen gas came from the water. It is unrelated to the oxygen that's in CO2. So that oxygen gas came from the water, and we have those hydrogen ions that came from the water molecule. Those hydrogen ions will be used to pump to one side of the thylakoid membrane, flow through ATP synthase, and produce a small amount of ATP. So that again is the same idea we saw in respiration. There's a small amount of energy that's released from an electron falling down the electron transport chain. That released energy is used to pump hydrogen ions across the membrane to make ATP. That is the same process we saw in respiration. Continuing on, we have that electron that was falling down this electron transport chain. Instead of oxygen being the electron acceptor, now we have something called photosystem 1, which is a collection of pigments. The definition of a pigment is a molecule that absorbs light. So light will excite a pigment in photosystem 1. When that happens, the pigment in photosystem 1 has an electron get excited by that light, and it again will get grabbed by a protein. So this kind of repeats what we saw on the left. We have an electron excited. It became excited due to the energy of light. When that electron gets excited, it again gets grabbed by a protein called the primary acceptor. That primary acceptor again passes down an electron transport chain. At the end of that electron transport chain, there is a molecule called NADP. And that NADP is waiting to pick up that electron. It picks up that electron and it picks up a hydrogen, which is the same hydrogen from water, the same hydrogen that got pumped across the membrane. So that NADP will pick up a hydrogen and the electron, and it will produce NADPH. That NADPH is storing an electron, it's carrying an electron. So just like we saw with the molecules NADH and FADH2 in cellular respiration, we have created a shuttle bus, which is an electron carrier. That electron carrier is holding energy in the form of that electron. So at the conclusion of the light reactions, we have produced some ATP, we have produced NADPH, we have produced some oxygen that is released as waste. The left and right side of the diagram here have some similarities and some differences. The next few slides show exactly this diagram, so I will quickly talk through those and then conclude with the Calvin cycle. So looking ahead, first couple steps, we've got our photosystem. Our photosystem is a group of pigments. Those pigments get excited when light hits them. Light is energy, so it gives the electrons in those pigments more energy. The excited electron gets grabbed by a protein called the primary acceptor. That primary acceptor is taking an electron, so we don't want that photosystem to run out of electrons, so water comes in to resupply the electrons at that photosystem. When that electron gets deposited at the photosystem, we produce hydrogen ions from the water and oxygen gas as waste. That primary acceptor then passes the electron down the electron transport chain. During that process of passing the electron, a small amount of energy is lost from the electron. That is used to pump the hydrogen ions across the membrane. It is the same process that we saw with cellular respiration. Chemiosmotic means we set up a concentration gradient where all the hydrogens are congregated on one side of the membrane. They won't like that because they're all the same charge and molecules want to spread away from one another. So those hydrogen ions will flow through ATP synthase to produce ATP. At the end of the electron transport chain, there is another cluster of pigments called photosystem 1. Those pigments absorb energy from light, causing electrons again to get excited. Those excited electrons are grabbed by a protein called the primary acceptor. This slide is showing what's happening again in respiration. It's essentially showing you a recap of what ATP synthase is. So we've got those hydrogen ions pumped to one side of the membrane. After that electron gets excited, this photosystem here will not run out of electrons because it's continually getting a supply of electrons from the electron transport chain. So we do not see water coming in to replenish the electrons again because there is a supply of electrons coming down the electron transport chain. After the primary acceptor grabs that electron, there is a second electron transport chain that ends in the production of NADPH. So we are making a high energy electron carrier. That electron carrier will get used in the Calvin cycle. So at the conclusion of the light reactions, we have made oxygen, ATP, and NADPH. We have used water and light. To conclude here is the Calvin cycle. You are required to know much less about the Calvin cycle than you are about the light reactions. That's just the way College Board has laid out the requirements for AP Bio. Kind of like how in cellular respiration, we didn't need to know a lot of details about the Krebs cycle. All right, so here we are, the second step, the Calvin cycle. The Calvin cycle will only occur because it uses what was made in the light reactions. So generally, plants kept in the dark will not grow because the light reactions are not occurring. We need ATP and we need NADPH to provide energy for the Calvin cycle to occur. During the Calvin cycle, 6 carbon dioxide molecules are glued together to make a sugar, a molecule of glucose. There are a variety of enzymes that rearrange those carbon atoms to create the sugar. You're not required to know those enzymes or know each step, so just the big picture here. We are using carbon dioxide, connecting the carbons to produce sugar. Lastly, to conclude this video, a reminder that the Calvin cycle does not use light, but it is usually going to take place simultaneously, because why would a cell sit around and wait if it can do something at the same time and not waste time? So the cell is going to try to make as much glucose as possible, so why not get everything going at once? So the Calvin cycle doesn't use the light, it uses what was made in the light reactions.